Okay, good afternoon. My name is Cliona and I'm from Cork Insttitute of Technology in Ireland. And I'm here with some colleagues who are also visiting us from Ireland. Here is Roy Sleator from Biological Sciences in Cork Insttitute of Technology, Bryan Kelleher from the Physics Department, Photonics in Cork Insttitute of Technology, and two colleagues from the University of Limerick, which is also in Ireland, Emmeline Pearson and Sarah O'Sullivan. Okay, so we're delighted to be here in Unicamp today and have the opportunity to talk to you. Thank you so much, Jose, Dr. Pisolato, for organizing this space. It's wonderful. So I don't know how much you know about Ireland. We know we have some students from Unicamp in Ireland this year on the Science Without Border program. So we're very happy with the students, and we're delighted to learn more about your university as well. Okay, so where is Cork? I suppose where is Ireland? Also, I'm not sure if there's a light. Okay, so here's Ireland, a very small country in Western Europe. Okay, a bit of a here's Ireland itself. Ireland, you may have heard of Dublin, the capital, which is here. Cork is here in the south, on the south coast. It's the second largest city. It's got a population of 200,000. What's important as well about the population is about a quarter, 25% of the population are students, because there are two big higher education institutes in Cork City, so it's a very important student city. Here are some photos of the city. It's quite an historic city. It's built on a very important port. And this map is something you see a lot in Cork. So the red area at the bottom is Cork, the county, and the rest is not Cork. Okay, here's some of the surrounding areas. Cork's on the south coast, on the Atlantic, so some beautiful landscape very nearby. The beaches are a little bit colder than beaches you might be used to in Brazil, but you can still swim in them, and maybe have a hot whiskey afterwards to get your blood circulating again. We've some castles. The Blarney Castle, it's one of the most famous tourist attractions in Ireland. It gets queues of people every year, because there's a legend to say that if you kiss a stone that's on the castle, you get the gift of talking a lot. And that's why Irish people talk a lot. So a lot of people come to Ireland to kiss a famous stone here. Cork is also really famous because it's the last place where the Titanic left from. This is the same place today. So today we get cruise ships, and there's a ferry ship to France every day from Cork. It's actually the second deepest harbour in the world, natural harbour. Also was voted one of the top 10 places to visit by Lonely Planet. And it was European Capital of Culture in 2005. It's a very vibrant culture. Okay, a little bit about the city. It's a very student-friendly city. It's Ireland's festival capital. There are a lot of festivals. There are about 24 festivals during the year. So in Cork, there's always a festival starting or happening or coming up. This week actually is one of the most famous festivals. The International Jazz Festival is on this weekend. St. Patrick's Day Festival is important everywhere in Ireland and places where there are Irish communities. I've heard that in Brazil, St. Patrick's Day becoming quite popular. Cork has great shopping and food, and the heritage and landscape is really nice. Okay, so just a little bit about Cork Insttitute of Technology. We have 14,000 students. About 50% of them are full-time students, undergraduate and postgraduate, and the other 50% are part-time students. So we have a lot of students who would work during the day and come to college in the evening to study, or maybe they could do 2 years full-time and then go to work in a company and come back and finish their bachelor's degree in the evening. We offer bachelor's degrees, masters, and PhDs. We have very strong links with industry, like Unicamp. Research and innovation is a very important part of it, in collaboration with industry as well. And we have a very varied curricula of subjects, especially science and engineering programs. And a lot of our programs are developed together with local industry. So apart from all the traditional engineerings, we also have kind of new novel engineerings that would mix maybe engineering and science for different niches in the market. We have 4 campuses. There are the 4 pictures here. So Bishopstown is our main campus, but we also have 3 other sister schools, which is the Maritime College, the Art College, and Cork School of Music. Also, Cork Insttitute of Technology was awarded Institute of Technology of the Year by the Sunday Times in 2010 and 2011. These are the programs that we offer. The Business and Humanities faculty and the Science and Engineering faculty. One of the highlights, I suppose, of the Business and Humanity would be the tourism and hotel catering. There's a whole installation for students to become hotel managers, restaurant managers. The restaurant they have, we can actually go and have dinner there. It's like a little company. The engineering is one of the broadest on offer in Ireland, and the art, music, and maritime. These are the master's programs, the Thought Masters. So these are 1-year Thought Masters, but it's also possible to do a Research Masters, which would take 2 years, or a PhD in all areas. What's distinct about Cork Insttitute of Technology is we've small class sizes. Typically there will be 20, maximum 40 students in an engineering science class. There's a very strong lab or practical. About 50% of work would actually be in a lab, and it's evaluated. Students go on work placement, do a final year project, often with the company where they did the work placement. And that company would often, when they choose the students for work placement, would be looking at investing in them for them to go back to work with those companies later. There's a lot of flexibility and choice, and we have 2 semesters, September to January and February to June. This is an aerial view of our campus. So we're on the edge of the city, about 10 minutes from the city centre. So most of the science and engineering are here. The sports facilities are here. The Rubicon Centre is here. I'll tell you a little bit more about that. The tourism is here. And this here is the student centre. So Cork has a philosophy that the student is at the centre of education. So the student centre is in the centre of campus. Okay, this is the central courtyard. So the students, all the student facilities are here. My office is up here. This is our library. It's actually a modern building, but the interior design is kind of in an old-fashioned way. It looks a bit like Hogwarts in Harry Potter when you walk in. This is the student center. This is the restaurant where students, one of the 2 restaurants where students eat. The music school is in the city center on the river. It's a beautiful modern building. Apart from, it's one of the most important centers for classical music in Ireland. And with the merge of the Institute of Technology, we've developed new courses. Apart from popular rock music, voice, theatre, there's also a whole load of industry-related degrees like music technology, sound engineers. The Maritime College is the only college in Ireland where you can study to be a Navy officer or a marine engineer, and it's in collaboration with the Irish Navy. It's on the harbour where the Titanic left from. The Art College is in the city centre as well. This is the building here. It's a very old college. It's 200 years old. It was actually the first college to have art and crafts and technical so it's vocational education in Ireland. So this is now part of the college. They have fine art, ceramic, photo. It's the only place as well in Ireland you can do a master's in art therapy. But there's also a range of multimedia, graphic, very modern technology mixed with art. We have an art gallery as well as part of the art college where there are exhibitions. We have a very active international student society, which is funded by the college. So they organize events every week and trips at the weekend for international students. Sports is also very important. Some sports you can actually get credits for. We invite students to do a module in sports and they get credits. Also we have a castle, which is part of the university. It's an observatory, so it's where our astrophysics group hang out. And it's also an interactive museum for the general public and children to get them interested in science at this side of the building. So if any students interested in doing a PhD in astrophysics, this is where they would study. Another highlight, I suppose, of Cork Insttitute of Technology is a Rubicon Centre. It's a business innovation hub where there's space for like 20 to 40 small startup companies, and they on this side of the building. And on the other side, we have a lot of researchers, and the researchers give the new startup companies advice. There's a resident entrepreneur as well in the building. And to date, 200 small companies have successfully developed from here and employed 2,400 people, a lot of who are graduates of CIT. This is a bit about our research. I won't go on too much because two of our researchers are here to talk to you today as well. This is Roy. A lot of our students enter international competitions. They do a lot of extracurricular project work. An undergraduate mechanical engineering student in 2011 won first place in a European competition. The second place was a master's student from the University of Cambridge, and third place a master's student from Heriot-Watt. So they have a very good practical skills during their education. Okay, these are some of the businesses that we work with closely in the region. These are students from Unicamp that we have in Cork at the moment. The first guy, Felipe, he's studying chemical and biopharmaceutical engineering. When he heard I was coming to Unicamp, he said, "Oh, take my photo and tell everybody I said hello." And there's another student, Bernardo. They're both from chemical and biopharmaceutical engineering. Okay, and we received 40 Brazilian students this year on campus. That was the day they arrived. Okay, I have a short video to show. I don't know how we're doing for time. Are we okay? Yeah? Although the sound, do I need to turn on sound?
Cork Insttitute of Technology is a leading higher education institution based in Cork City on the south coast of Ireland. We offer a wide range of flexible full-time and part-time courses at all levels, up to and including PhD. The main campus is located in Bishopstown, Cork. The Crawford College of Art and Design and the Cork School of Music are both located at campuses in Cork City Centre. The National Maritime College of Ireland is located on the shores of Cork Harbour in Ringaskiddy. In our Faculty of Engineering and Science, we have a long tradition of providing programmes of study, research and other services in the areas of science, technology and engineering. CIT is in fact the second largest provider of engineering education in the country. The Faculty of Business and Humanities delivers an equally wide variety of career-focused business and humanities courses and has a proven track record in the successful provision of ongoing professional development. The CIT Crawford College of Art and Design, located at the Sharman Crawford Street campus in Cork City Centre, runs a range of programmes in art, multimedia, new media and other cognate fields. The CIT Cork School of Music provides third-level qualifications in music, music technology and theatre and drama studies from a state-of-the-art city centre facility overlooking the River Lee. The National Maritime College of Ireland, based in Ringaskiddy on the shores of Cork Harbour, is a modern purpose-built facility developed as a consortium between Cork Insttitute of Technology and the Irish Naval Service. Programmes are offered by the college across a range of nautical fields. Cutting-edge research takes place in the Institute across and between a number of disciplines. Our research is enterprise-focused and solution-orientated with a strong emphasis on collaboration and knowledge exchange between CIT and outside enterprises and organisations. CIT is home to the Rubicon Centre, Ireland's premier business incubation centre, which is home to over 40 knowledge-based startup companies. The Institute offers an array of student services, clubs and societies. Our sporting and leisure facilities are among the best in the Irish higher education sector, and our students also have access to a wide range of other student facilities, services and supports. CIT students experience vibrant and diverse campus life. Our staff are dedicated towards our students' personal and social, as well as their academic and professional development. Our graduates leave CIT ready to take their place as professionals and practitioners, as members of their local communities and as citizens of the world at large. Cork Insttitute of Technology. Knowledge at work.
Okay. Thank you very much. I have some leaflets here if anybody would like to take some, general and on the master's programs. And I'll hand you over now to Roy from the Biological Sciences Department.
Thanks, Cliona. My name is Roy Sleator, and I'm a lecturer at the Department of Biological Sciences at CIT. So hopefully what I'm going to try and do is, in a very short time, give you a brief overview of some of the research that's going on in the Department of Biological Sciences. So I'm going to start by kind of outlining, I suppose, some of what we call platform technologies. In other words, where is our area of expertise? And within that, maybe highlight a couple of case studies that I'll talk a little bit more in detail about. So I suppose one of the first areas of expertise within the Institute and the department is genomics. In other words, looking at the genomic or gene content of organisms. Outside of that, metagenomics, which I'll talk about, that's highlighted because it's going to be a particular case study. We also have an interest in 3D protein structure prediction. This is in silico or computer-based structure prediction modeling. And we're also very interested in phylogenetic analysis. So again, focusing on evolution of particular genes in the first instance and also proteins. So the next field or area of study or expertise within the department is looking at bacterial stress responses. So this is how particular bacteria, pathogens, those that cause disease, and also probiotics, the so-called good bacteria, deal with stresses in their environment. Okay. One of the environments which we have a particular interest in CIT is the human gut or the small intestine, and how bacteria, both good and bad, can survive and multiply in this fairly harsh environment, and specifically the genes and proteins that allow it to do that. So again, bacterial stress response and probiotics are highlighted, and I intend to talk a little bit more detail on both of those topics. Next section is a little bit more different to the previous two, which is called pathobiotechnology, and this is the genetic engineering concept or approach that we're involved in CIT. More specifically, what that means is rational design of improved pharmabiotics and novel biocontrol strategies. So taking our knowledge of bacterial stress responses and our knowledge of bioinformatics and genetics to try and engineer improved bacteria and improved methods of dealing with the so-called bad bacteria or pathogens. So the first case study that I'm going to concentrate on is metagenomics. And again, for those of you who haven't heard of metagenomics before, it's essentially an extension on genomics. So metagenomics looks at particular environmental niches, be it soil or the ocean or a termite or the gastrointestinal tract. And it involves extracting all of the organisms or microorganisms associated with those environments. Okay. So once we have all of the microorganisms extracted, then we extract the DNA, the total genomic content of those environments, with the idea of mining that information. So this is a very powerful technology because for the first time, from a microbiological point of view, we can't culture quite a lot of these, quite a big proportion of these bacteria and viruses can't be cultured in the lab. But we can look at their DNA, and if we know what genes they have, we can predict what proteins are expressed. So again, it's a very powerful technology. As I said, in CIT, we're particularly interested in the human gut. And so the metagenomics approach that we've taken in this instance is mining the human gut. So how do we go about that? How do we go about looking at the human gut metagenome? Well, rather disgustingly, what we have to do is take fecal samples from individuals. So a stool sample from an individual. In this case, it was a 26-year-old Caucasian, in other words, postdoc in the lab. Postdoc will go to any extent to impress their supervisors in CIT, and giving their stool samples is one of those. So once we have the fecal matter, we go about extracting the DNA from that microenvironment. The DNA is then broken down into fragments of about 40,000 base pairs and cloned into cloning vectors. And these cloning vectors are then transformed into a cloning host, in this instance, an E. coli host library. So from this point on, we take one of two strategies. The first one is a sequence-based approach. And in a sequence-based approach, we look at the sequence. So we interrogate the sequence that comes back. Okay. This is a very nice approach, but it also generates a huge amount of data. And as biologists, we're not altogether comfortable all of the time dealing with huge amounts of data. And so the sequence approach has its limitations in that respect. Another approach, which is more direct, I suppose, is the so-called functional approach. And in the functional approach, we identify the gene systems based on the proteins that are expressed. So we're looking for a phenotype, a physical appearance of the expressed genes. So for example, take the example of salt tolerance, the ability of bacteria to survive in very salty conditions or conditions where there is no free water. And the functional screen involves plating them onto media which is a high salt concentration, which would not normally allow the bacterium to grow. So let's concentrate a little bit on the sequence-based approach firstly. So as I said, biologists were not necessarily that comfortable dealing with huge amounts of data, but we're now in the post-genomics era where huge amounts of data are forced upon us. And so one of the collaborations that I was involved in CIT is with our computer science department. And of course, the computer scientists are very comfortable working with large amounts of data and writing algorithms and programs to be able to deal with large amounts of data. The schematic here represents a workflow which was developed by one of our PhD students, a joint student between computing and biology. And John, who's the student's name, essentially wrote a program which will take the sequence data and pass it through a series of workflows to get down to the essence of the genes that we're interested in, the proteins that we're interested in doing. So this is, I suppose, an interesting story from another point of view, and it follows up on what Cliona mentioned about our innovation center, the Rubicon, because this project, which started out as an MSc project, led to intellectual property, which resulted in the development of a spin-out company. And so this software is now licensed, and the MSc student was actually the first employee of our spin-out company. And so this is generating revenue because it solves a very acute problem within the biological sciences. So you go from billions of nucleotides of information to a very nice genomic map. And this is just an illustration of the type of feedback you get. So showing the relative location of the genes, their directionality, codons, promoter sites, and so on. Okay, and so this is all done in an automated fashion. So again, this is a project which resulted from a very nice collaboration between computer scientists and biologists. And I think that's a key area of strength within CIT, that we have a very interdisciplinary approach to solving complex problems. So in this instance, it was a complex biological problem solved with the aid of computer scientists. But this type of approach is a model that we have taken within the Institute to solving all different types of problems. Functional screen, on the other hand, involves a more robust approach looking at hardware as opposed to software. So our library of clones, each containing a different fragment of our human gut metagenome, were plated on these 386 well plates, and we had hundreds of these 386 well plates. And as I said, the functional approach involves picking each individual colony and putting it onto a different stress selection medium. Luckily enough, within CIT, we have a suite of robotics which facilitates the creation of these libraries in these plates in the first instance, and also the screening of the library. So traditionally we use PhD students, no longer. We now use very advanced robotics. So to cut a very short, or a very long story short, this approach involved the screening of 23,000 clones. So each individual clone had to be replicated onto different plates. So a very laborious approach if you were to do it manually. But again, because we have automated systems, we could do this in a matter of days. These individual clones were then incubated under conditions of osmolarity which would not normally be permissible for growth for the E. coli strain that we were using. And so any strains that did grow were because of the genes that were cloned in there in the first instance. And remember, these genes came from the human gut microbiome. Again, we identified 53 possible osmotolerance clones, and this table here is just a snapshot of a few of them. In fact, these are the ones you would have nearly expected to come back when you blasted them against the NCBI database of known gene systems. They were all in some way linked to osmotolerance. But what I haven't mentioned on this slide is that we identified 5 genes which are completely unique to osmotolerance. These are genes which look like nothing else on our databases and are a brand new mechanism for protecting bacteria against low water environments. And of course, again, from a commercial viewpoint, these particular gene systems and their encoded proteins can have very beneficial applications in terms of drought tolerance and engineering plants for drought tolerance and so on. Okay. So again, that's feeding into our IP pipeline and business model. So having identified new stress genes, new genes with encoding proteins which allow bacteria to survive in environments which they would not normally survive, what do we do next? What's our next kind of objective, I suppose? And that leads into this idea of pathobiotechnology. Now, I'm guessing most of you haven't heard of pathobiotechnology before because it's a name that I made up. In fact, I made it up with my supervisor at the time, Colin Hill. And essentially, the catchphrase here explains what it means. We're using bad bugs to do good things. So in this pathobiotechnology concept, if you think of a trait which allows a bad bacterium to be bad, and if you take that trait and express it against a good bacterium, then in essence, it can help the good bacterium to be better. So what am I talking about in terms of specifics? Well, if you look at a pathogenic trait, a trait which allows a bad bacterium to be bad, for example, resistance to environmental factors, the ability to survive passage through the stomach, for example, the ability to survive within the acidic environment of the stomach. From a bad bacteria point of view, that's a bad thing because it allows the bacterium to get through the digestive juices and establish an infection. However, if you're trying to engineer a probiotic, a good bacterium, to survive the stoic acid, then in that context, it's a good thing. Okay? So panobiotechnology involves looking at what we've learned from our studies of pathogens or bad bacteria and applying that to the good bacteria, re-engineering improved probiotic strains. Okay? So at CIT, our model pathogen or model bad bacterium is Listeria monocytogenes. This is a gram-positive bacterium which is linked to spontaneous abortion. So it's a pretty bad bacterium. Our good bacterium, Lactobacillus salivarius, is a commercial strain which has been shown to have probiotic qualities. Okay? And a study recently published in Nature actually shows that Lactobacillus salivarius protects against Listerial infection. Okay? So again, keeping salt stress or low AW stress as our indicator stress, work in our lab led to the identification of 3 gene systems which encoded 3 proteins in the membrane of the Listeria, which allow it to take up these protective compounds, betaine and carnitine. So when the cells find themselves in a low water environment, these uptake systems take up these protective compounds which protects the cell from plasmolysis or shrinkage and death. Our probiotic, our good bacterium, lacks these uptake systems, and so at low AW environments or in the absence of water, no protective compounds can go in, and so you get plasmolysis. The cell dies. Okay? So our question was, if we take any one of these proteins, these protective proteins from the bad bacterium, and express it in the good bacterium, can we now improve the ability of the good bacterium to survive? Okay? And so that's exactly what we did. We cloned the smallest of these promoters, beta, into Lactobacillus salivarius, and hey presto, it took up the protective compounds under the stress condition, and it protected the bacterial cell. So using a genetic engineering trick, we have now got, albeit a genetically engineered probiotic, but a probiotic which can survive highly stressful environments. Indeed, when we tested it against a number of different stresses that you might find in the food environment, for example, 7% sodium chloride, our transformed or our genetically engineered strain grows much faster and better compared to its wild type equivalent. This is the one which lacks the beta gene. So that's at high salt. Indeed, at low temperatures that you would expect for frozen foods, for example, again, our transformed probiotic strain survives significantly better than its wild type, which dies very quickly. Okay. Another stress which is interesting from a food process point of view is barotolerance or high pressure processing. So a lot of foods are subject now to high pressure processing to kill the bad bacteria. But of course, this also has the effect of killing the good bacteria. Our good bacteria, as you can see, survive significantly better under high pressure conditions relative to the wild type. So these are in megapascals. So just to summarize, I suppose at this point, we now have engineered probiotic strains which can survive in the food matrix. Okay, so you're probably all familiar with this particular probiotic drink, which I won't mention. So the next step was to determine, well, if they survive well in the food, how well do they survive in the gastrointestinal tract, in the animal model, okay, within our gastrointestinal tract. And again, we get some really good news here because the open circles here represent our transformed probiotic strain, our genetically engineered strain, relative to its wild type. So this is fecal pellets taken from animal models. So the animal model we use is the mouse. So the mouse was fed with the genetically engineered probiotic strain, the wild type, which was not the genetically engineered strain, and we extracted fecal pellets or stool samples every 3 days, for a period of 33 days. And you can see here that the genetically engineered probiotic strain was present at higher concentration in the stool, indicating that it was present at higher concentrations within the gastrointestinal tract. So not only have we engineered probiotics which can survive best in the food, but they can also survive really well within the gastrointestinal tract. So moving away now from food science and into the idea of clinical medicine, we have potentially a probiotic strain which survives for longer periods at higher numbers in the gastrointestinal tract. So what effect does that have against infection? You might have remembered that I said earlier that our good bacterium, lactobacillus, fights off the bad bacterium, Listeria monocytogenes. So our next question was, well, does our genetically engineered lactobacillus fight off Listeria more effectively than the wild? So at this point here, on day 15, you can see that there's a difference between the wild type and the genetically engineered one. We fed these animals, these two mice groups, with Listeria, the bad bacterium. And again, this is an example of a kind of collaboration. We were able to engineer a Listerial strain that gives off light. And we have an instrument in the institute which measures the level of light in the live animal. So the animals don't have to be killed. We can see the amount of light coming through the body of the animal, and that amount of light is directly proportional to the amount of infection. So where you see light, you see infection. Where there's no light, there's no infection. So the plate here, you can't see it very well, but on the top there is the spleen, and on the bottom there is the liver. You can see quite a lot of light coming through here, represented by blue, and a lot of light coming through here, represented by blue and red. These are the controls. This is the genetically engineered probiotic, or the liver from the animal, which was fed that. You can see that there's no light there. So no light means no infection. No infection suggests that our probiotic has worked really well. So we've gone from a food science or food engineering to now a clinical medicine application of our engineered probiotics. So this has led us to the next phase of our study, and again, this is in collaboration with the Department of Medicine in the nearby university, where we're going to look at using these genetically engineered probiotics as vaccine delivery platforms. So instead of injecting a vaccine into an individual, feed them the probiotic, which will express the vaccine. So no needles, which again is a huge advantage in third world countries, for example. So indeed, we've engineered our probiotic strains to get out of the gastrointestinal tract. So we've given them proteins which allow them to extricate or remove themselves from the gastrointestinal tract and enter into the intraperitoneal cavity, where they're then taken up by macrophages. These are cells of the immune system. Once in the macrophage, we can engineer them to remove themselves from that environment as well. And ultimately, what we get here, and again, this is immunology now, so a different field of expertise again, what we get is a large and boosted stimulation of the immune system. So in essence, we have created a strain which can immunize against subsequent infection. One of our target pathogens in this study is Mycobacterium. And again, you're probably all aware of Mycobacterium as an extremely dangerous pathogen. And again, using our genetically engineered probiotic strains, we find that there is less light when they're treated with our strains, so less light, less infection, relative to the control. Okay. So that kind of finishes the science. Now you're probably wondering where all of this science goes on. Well, Cliona alluded to the fact that we have a new building. We have several new buildings, but this is one of our new buildings, which we call the CREATE Centre. This is an acronym for Centre for Research in Advanced Therapeutic Engineering. So this is a two-story building which houses three different research groups. So there's biological sciences and chemistry in the BioExplore Research Centre, a medic, which are biomedical engineers, and then finally physics. So Bryan will speak to you about some physics research that's going on. So the idea behind CREATE is if you house these different groups in the one area, one coffee room, we'll all start talking, and new ideas will be generated, and new approaches, and hopefully I've illustrated some of these interdisciplinary approaches and some of the advances that can be made from that. Again, this is a flow of the different types of things that are going on within CREATE. One area which is very close to my own heart is bioinformatics. So again, the fusion of computing with biology. And indeed, this bioinformatics is an important interdisciplinary platform within Cork Insttitute of Technology. Indeed, we've just set up a taught master's degree program which focuses on bioinformatics or computational biology. This is a one-year master's. The course code is here. By clicking on the CIT website, you'll be able to get to the different courses. And for any of you who might be interested in that, any opportunities which arise from that course, you can always just email me. So my email address is there. I'll leave some business cards at the end and people can track me down. But again, this is a one-year master's, which is a taught master's, and provides a brilliant stepping stone for those who want to put their foot in research for 1 year, progress onto a PhD or enter into industry. Again, the major, I suppose, vision of this computational biology master's is that it brings together different expertise and adds value to your primary degree. So that's all I have to say to you. Again, I'll leave some business cards at the end, and if anybody would like to ask me any questions, I'd be more than happy to talk to you. Okay? Thanks very much.
Okay, so thanks again for allowing us to present some of our research here. I'm Bryan Kelleher. My name isn't here, but it will appear later. I'm just going to talk a little bit about photonics research and CIT in particular, and as well how photonics research in Ireland sort of operates and the relationship between all the different institutions. Okay, so very simply, what is photonics? So often it's described as the science of generating, controlling and detecting light. Essentially, anything that requires light in one way or another, it can be considered as being part of photonics. And quite often it's seen as an enabling technology. So by this, what I mean is that photonics on its own has its own technology, but as well many other technological areas require photonics in order to function well or function better. So very obvious ones are communications, for example, optical fibers, and even for using the internet, or then we have life science and health. And in fact, we just saw in Roy's talk a second ago how they use detection of light in order to detect some infection. So it's another application in which photonics can appear. Energy and lighting is a very important one as well. Solar cells in particular is something that's obviously of great importance today to try and use energy in a more efficient way. And finally, security and metrology. So the kind of standard example is gas detectors. So if you wanted to make sure that in some factory carbon monoxide wasn't going to kill everybody on the floor, well, one way is using light in order to detect the particles in the air. This is our group. Well, it's a slightly out-of-date photograph of our group. This is our group leader here, Dr. Guillaume. This is me shining out here at the back. This person here actually was a postdoctoral researcher at the time with us, Christian Bonato. He is from Brazil himself. He's currently a lecturer in Pelotas. So our centre is known as CAPA, the Centre for Advanced Photonics and Process Analysis. We're based, of course, in CIT primarily, but a large percentage of the group also works in the International Institute, which is an institute in Cork City Centre, again a sort of multidisciplinary institute, but photonics is a big part of it. At the moment, this is our staff number. We have 16 researchers, 7 PhD students, and these numbers are constantly fluctuating up and down. These are some of the highlight figures. We had, in the last 5 years, more than €6 million in funding, most of which probably came from the EU. More than 35 projects, and this combines national projects, so projects where we just do the work ourselves or in collaboration with other institutes in Ireland or across Europe as a whole, and many industrial projects as well. And in the last 5 years, we've published more than 55 peer-reviewed journal articles. These 4 categories kind of describe what the main activities of CAP are. So the first one is, of course, the most important one in some ways to us. Certainly to me, it's the research area. And for any prospective students, this is the one where they would work. The others are more at the postdoctoral level, but we have many strands within the research. We focus a lot on semiconductor light sources. We use these lasers typically to build complex systems then, and these can be used to study nonlinear dynamics, and it turns out that these have lots of analogies in many other areas, not just with lasers, but in population, networks for communication, and so on. We now also have a large area of product development. Again, started off with semiconductor materials. Illumination for industrial applications is one of our main ones as well at the moment, and we also have one for testing the purification and helping to purify water. Process improvement. So we have various things, I can say more about them later, but we use spectroscopy for process control, analysis of products and processes. For example, some multinational corporations might come to us and they, kind of in collaboration with the consultancy part, and say, oh, we have such and such a process. It doesn't seem to be very efficient. Is there anything you can do to help? And this is a growing area for us. So within, especially the research area, these are our capabilities. So the capabilities that are in our own actual labs. The first one is laser dynamics. So this is kind of fundamental science one. The analysis of nonlinear dynamics, semiconductor lasers, what happens, for example, with feedback. If this laser is in some network, some of the light reflects back into the laser. Does it make it all go crazy, or does it all stay fine? Some spectroscopy capabilities. One in particular, which is called two-color pump probe spectroscopy. The details aren't really important, but it helps you to analyze what happens within a material over very short time scales. Similarly, time resolved photoluminescence. We have several imaging capabilities, Fourier transform, infrared, and Raman spectroscopy. We have some nice equipment for scanning electron microscopy, and lately as well we've started to do a lot of optical design and modelling, so using in particular the package ZMAX. So those were the capabilities within our own lab, but based in Tyndall Institute we have access to a lot of other capabilities as well that are shared amongst all the different groups within the Institute. These are just three of the examples. There's a lot of growth of semiconductor material, so this is a photograph of one of the reactors, as well as fabrication. So what's happening here is the wafer of some semiconductor material is being spun out, and several clean rooms and packaging and photonic integration circuits. So it's part of the reason why part of the group is based in Tyndall. It makes it very attractive that you can really start off with the atoms and work your whole way up, grow some material, fabricate it into something, and then finally package it. So this is the logo, or this is kind of motto for Tyndall: from atoms to systems. As part of the national scene, these are probably the four major cities: Dublin, Galway, Limerick, and Cork. And each one has several photonics research. So in Cork, there's us, of course, in CIT and Kappa, and then there's groups in Tyndall and in the university. In Dublin, several: Trinity College, Dublin City University, Dublin Institute of Technology. Galway has a lot of photonics research going on now as well, but it's much more applied. And in Limerick, we have some collaborators as well in their materials and Surface Science Institute. Probably you'll hear a bit more about Limerick in a few minutes. So this is a new centre which is based in Tyndall. It's called the Irish Photonic Integration Research Centre. It's a new project we just started this year. It got a lot of funding from the government. There's 16 industrial partners based on multinational corporations, small and medium enterprises, and several startup companies as well. Tyndall is coordinating the project along with us, and University College Cork are also part of it, and then DCU, the Dublin City University, also have some researchers involved in the program. So the outline of it is there are 10 principal investigators. These are kind of their headline figures. Many publications, plenty of collaborations, money, patents, and so on. Again, it's sort of divided up into the four things. Starting off with materials, turning these into devices, packaging them, and then putting them into systems eventually. And we got some nice review. One of the reviewers said he would consider it as being of the best in world class. I said I better include that. Another program throughout the country is the Inspire program. It's an integrated science platform for Ireland. So most of the institutions, most of the higher level institutions are involved in this. There were 2 phases. The first phase was where we were given a lot of money and told to buy world-class equipment, put these in the labs and get the facilities in place. And then the idea would be that everybody would be able to share these facilities later. As Cliona showed earlier, the size of the country isn't actually that big. It's probably comparable to Sao Paulo State, so it's not that big a deal if somebody down here wants to travel up here to do something to use some of the facilities that were funded here. The second phase is the graduate education program then. I'll go through it again in a sec. So like I said, the first phase, lots of money, 3 particular areas: bio-nanoscience, nanoelectronics, and our own area, nanophotonics. And these were some of the machines and facilities that were put in place as a result of it. So now these are being used in phase 2, which is a structured PhD program. So somebody can apply for a PhD, do it in a structured way so that they do research, but they also access a lot of different modules. And everybody is sharing the program in the involved institute, so their module delivery, that can be local lectures, video conference and web download, and all these modules are shared amongst all the different institutions. These are examples of some of the modules. Everybody takes at the very start an introduction to nanoscience course, which is a week-long workshop where all the students get to meet each other and get to know each other in lectures during the day and whatever they want that night. There are several technical modules, of course: photonics, electronics, nanobioscience, and then a large part, a very important part of it, are transferable skills. So there are very good modules in communications, presentation, management, entrepreneurship, and so on. And a lot of these transferable skills are really led by industry. They tell us what they want the students to come out with, and that's what we try and give the students. This just gives an example of some of our collaborators academically, internationally. Of course, one of these red dots covers the whole of Ireland, as you can see. And then many around Europe, one in India. We have UCLA in Stanford in the States. You see there's a big gap down here, so we're hoping that as part of this we'll find something down here. And who knows, maybe end up with some nice collaboration with Unicamp as a result. These are several positions that are in our group alone at the moment. As part of the Science Without Borders, we have several positions. There's one advertised, but really, if the student contacts us and seem interested, then we can always find a project. There are 2 projects that are currently advertised on findphd.com. So these are funded by Europe and by the Irish government. First is a joint project with UCLA, Queen's University Belfast. And the second, and it's about nanopillar lasers. So it's a new type of growth to develop new lasers that should be a lot more energy efficient than current technology. The second is photonics for biomedical applications. And also, the Institute has a program called the RESAM Scholarships, and these are open to any discipline. Somebody just goes to the website, they get a list of all the different groups and centres within the institute and contact details, and then they contact the person that they think they might be interested to work with, and if they get on together, they can write a proposal together, and it's submitted, reviewed, and possibly leads to a PhD. So that's just summarising some of the things I said. Good facilities, good researchers, nice integration around the entire country, a strong network of international partners, and I just say, don't be shy. If anybody would like any information or is interested or anything, then you're probably better off just contacting me, but either of these addresses will do. So if anybody does have any questions, I'll be here afterwards. Quite welcome to come up.
Hi everybody, bom dia. Muito obrigada pela oportunidade de falar com vosotros. I obviously don't speak very good Portuguese, but I speak very good Portuñol, so a mix of Portuguese and Spanish. I have the presentation for you today in Portuguese, so I'm going to talk to you in English, but just to give you a sense of, and possibly more what I'm looking at today is the undergraduate opportunities that we have for Science Without Borders at the University of Limerick. So I've already heard from Cork, and if you travel maybe about an hour and a half down the road, you end up in the University of Limerick. My name is Emmeline Searson, and I'm the manager for the programme at the University of Limerick. And I'm here as well with Sarah O'Sullivan, who's our representative in Brazil. She's based in Rio de Janeiro, so she'll be presenting or introducing herself to you at the end. So just to give you an idea of where we are. So this is this is Limerick. It is the third largest city, or sometimes they say Galway, sometimes they say Limerick, but we're definitely after Cork when it comes to size. And as you can see, this is the city, and the university is located 3 miles outside of the city. So it's very much a self-contained campus. It's very, very beautiful. I worked there for a number of years, and I still enjoy working there every day. We have a river running through the campus. It's kind of like being in a wildlife park sometimes. We have swans, we have ducks, we have beautiful trees, all those kind of things. So a lot of our students, we asked our Brazilian students to tell us what they thought, and a lot of them were like, it's so green. And it's very green here, but it's very, very green all over Ireland. And one of the nice reasons, or why do we have such a beautiful green country, and one of the reasons why we have such a beautiful green country is because it rains. So this is very, very important when you come to Ireland, that we have a very different climate. But then a very interesting culture that goes around the whole idea of different type of weather. So that's something to consider as well. So here we are. This is the River Shannon, which is the longest river in Ireland, and it travels all the way through the city and then out to the university. So it's important to point that out because it makes it's a very focal point of the city and the university. A lot of riverside activities, cafes along the river, and then obviously into the university we have rowing, we have windsurfing. Sports is very, very big at the university as well. And obviously where we're located, we're only 30 minutes from Shannon Airport. So it's very, very easy for you to travel around Europe, so not only to experience Ireland, but also to experience Europe as well. And this just gives you an idea of the flights that we have from Shannon and then from Dublin. So plenty of opportunity for you to discover Europe and Ireland. Some kind of images of what it looks like, to give you an idea. Lots of castles. Which a lot of students, we have a lot of on the undergraduate program, we have a lot of American students who come to Ireland as well. So students from Japan, from America, from China, and obviously we have students now from Brazil to Science Without Borders. So a huge mix of students coming to study at the university. These are real photographs. This is what I like about them. They're not made up, they're real photographs. It actually looks like this. Just to give you an idea of kind of the idea, so you have a sense of what it's like. Obviously, as you all know, music and culture and the pub is very big in Limerick and Ireland as well. And we have, on campus, we have the Irish World Academy of Music and Dance. So students coming through Science Without Borders can, for example, study engineering. We have aeronautical, mechanical, biomedical, but they can also study Irish language, Irish folklore, Irish dance if they want to do that. And that's adds a very kind of interesting dimension to their experience in Limerick. This is the city. So it's a small city by Brazilian standards. It has 80,000 people, so probably the same as Unicamp, being the whole city of Limerick. And sports is very big. You'll see here is Thomond Stadium, which is where we play rugby. So this is a lot of our students go to matches and they enjoy all that. And we, as I said, we have an undergraduate program in sports science or kinesiology on campus. So a lot of professional athletes will train in the gym. So you could be, working out next to a rugby player, a professional rugby player. So, again, an interesting dimension to the program. The city of Limerick is interesting in that it has, for example, you'll see a lot of modern buildings here, but also we have a 13th century castle in the city, King John's Castle. So a huge amount of history around the city. very interesting in terms of, you know, learning about, some people have seen the film Angela's Ashes, which was set in Limerick, and there's a cultural tour around that as well, so a lot of different things to see and do. And this is the campus. So as I said, we have a huge amount of space, and this is what it looks like. Okay, 12,000 students. So again, much smaller than Unicampi. 9,000 of those are from our undergraduate, and the rest are postgraduate. We also have, again, huge sports activities. We have a 50-meter swimming pool, and as part of your program, you would also have membership of the gym as well. It's a young university. It was created in 1972, so we just celebrated 40 years. Extremely dynamic, and we have a very strong co-op program, so quite a very strong focus on employment. And obviously, another important part for you to know as well is that you live on campus. So we have 5 student villages, and all our international students on the study abroad program will come and live on campus in apartment-style accommodation. You will live with Irish students or other international students, so you have to learn to cook with them, to speak a lot of English. So it's a very, very integrated program when you come to Limerick. Just to give you an idea of some of the images of the campus, so this is our pedestrian bridge, which is the longest pedestrian bridge in Ireland, and was built by the Eiffel Company, so the Eiffel Tower Company, the same company. This goes from one county, so we're actually in County Limerick and County Clare. Our library, we also have 1,000. They're available to you, and they also organise activities at the weekends for you to do as well. So there's plenty of things to keep you busy. You can see you have your own study section as well in terms of the apartment, and obviously here living with other Irish students as well. Again, as I mentioned, sports is very, very big in Limerick. This is an Irish sport called hurling, which is the fastest field game in the world. And you will learn, when you come for orientation, we actually teach you how to play that. So a lot of students enjoy that. Gaelic football, basketball, track, and obviously rowing is very, very popular. We have a boathouse on campus as well, and an ergometer as well that you can practice on. So just to give you an example of some of the 4 faculties that we have. So we have science and engineering, which is our biggest faculty, business, education and health sciences, and then arts, humanities and social sciences. As I mentioned earlier, in terms of engineering, aeronautical engineering, biomedical, mechanical, we have product design, we also have equine science for students. Business was voted the best business school in Ireland last year. Courses in accounting and finance, economics and finance, entrepreneurship. And as I said, if you can mix, you're not in any way restricted to one faculty. So if you're an engineering major and you want to take some modules here in humanities, you can do that. So it's very much about building your own type of program that you want to do and trying out different things, as well as meeting the requirements around your major as well. Education and health sciences, we also offer courses in nursing, and they're all available to, most of the modules are available to international students as well. Architecture, which is very, very popular. We currently have 5 students from Brazil studying architecture in Ireland, and actually one of our lecturers studied here as well. So this is very, very much design studio focus, so a lot of very popular courses with our students. These are our civil engineering students. The interesting thing about civil engineering, they built this, and they obviously did a good job of it, is that they use PBL, so problem-based learning. Problem-based learning is very much focused around group work. You're given case studies and you're then presented and have to work out the problems yourselves in your groups. So again, a very good course in terms of interacting with other Irish students, using your English and really learning a different type of pedagogy that you would maybe not be used to as well. Our aeronautical engineering students making one of their successful planes that flew, which is also very good. And they did enter this competition every year. So as I said, mechanical, aeronautical and biomedical engineering very, very strong as well. And then just to we mentioned things like, integrated accommodation, but also what's very important is, as I mentioned, you can put the program together yourself, but you also have 2 weeks where you can actually try out different modules. So if you try, for example, a module in mechanical engineering that is too difficult, you can change. You can try something in aeronautical. So there's a huge amount of flexibility on the program in terms of the courses that you can put together yourself. Most students on Science Without Borders at the University of Limerick are taking 3 courses in the major, 1 kind of Irish music or dance, and then an English language class as well. So it depends on your own requirements in terms of your English language level, etc. Is there anything else that I wanted to mention to you? We also have a volunteering program, the President's Volunteer Award, which allows international students to volunteer in the local community. And that goes on to their transcript. It's only 12 hours over the semester. That's really 1 hour a week. But again, another really good opportunity for you to integrate into life in Limerick and to, to engage with the local community. So it's a very, very popular program with both Irish and international students. We also organise field trips for you, so 2 field trips per semester. We have a GPA system, the same as in the US. And then also we have most credited 60 CTS per module. So the course load for most students is 5 modules, as I mentioned. And just to kind of give you a very brief idea, I mentioned some of the other extra courses that you can take outside of your major: sociology, history, Irish language, Irish dance. At the end of the semester, we have a performance where all the dancers and musicians come together and they show us what they've done. So it can be very, very interesting. You can imagine Irish American students or Japanese students giving their slant on Irish dance, and we're hoping to have some interesting things with our Brazilian students at the end of the semester. Extracurricular activities are very, very important again, and just to give you an overview of the other courses that we're offering. Games development is very big as well. I mentioned architecture. Software engineering, we also have a research centre on campus in the area of software engineering, which is very, very big. We have languages, then literature. We teach Spanish, Japanese. Again, if you want to take a beginner's class in any of these, you can do so as well. And then in terms of other activities that you can get involved in, clubs and societies, at orientation, these clubs will come and talk to you about what they offer to students. So a lot of students will do the Outdoor Pursuits Club, which is a society which basically travels around Ireland, does hill walking, and they offer trips at very low cost. So it's a very good way for you to see Ireland. And again, it's all about meeting Irish students and integrating and, not making the most of the time that you spend in Ireland. The International Society, as Cliona would have mentioned for CIT, it's the same in Limerick. We have a very, very strong international society, and we would recommend that all students would join. They're really good at helping you to integrate and organising activities for you as well in the evenings. Just to give you a little bit more on the accommodation, these are our 5 student villages. All own room, they all have the internet access as well. So really top class. Our accommodation was voted the best last year in a recent student survey. That's a very, very quick overview. I know we're kind of tight for time, but if you have any questions, feel free to email us at study.abroad@ul.ie. Sarah's going to introduce herself very, very briefly to you as well. And remember that Sarah speaks Portuguese, so any questions you're welcome to send them to us in Portuguese, and we'll do our best to answer them for you. So thank you very much for your time, and if you have any questions, we're happy to take them.
Alô, boa tarde. Eu sou Sarah Sullivan, eu sou do Galway, que é mais em cima, um pouquinho mais em cima no Irlanda. Eu estou morando aqui no Brasil há 5 anos, no Rio de Janeiro, adorando o seu país. E com muito orgulho estou vendo que tem essas ligações agora entre nossos 2 países. Lá na Universidade do Limerick já temos 35 estudantes brasileiros de 13 estados do Brasil. E o que eu acho muito interessante, a Universidade do Limerick, eu estou trabalhando com eles agora, eu comecei esse ano agora. Eu acho que é muito interessante, eles têm uma brasileira que está trabalhando lá, fazendo apoio para todos os estudantes que estão lá. Então ela faz, Michelle, ela faz uma reunião com as brasileiras toda semana para ver se eles têm dúvidas, algum problema de adaptação, porque nosso país, nosso clima é diferente. Durante o inverno, os dias são muito curtos. Você tem pôr do sol 4h30 da tarde. É escuro. Mas no inverno, durante o verão, você tem pôr do sol 11 da noite. Meia-noite ainda tem sol, ainda tem luz. Então, é um país maravilhoso, com muitas coisas. A cultura está maravilhosa, música, tudo. O Limerick é uma cidade muito linda, castelos antigos, tudo a ver lá. Eu acho que a gente está fazendo muita coisa para receber vocês e eu vou deixar meus cartões aqui e se vocês têm alguma dúvida, seria um grande prazer para ajudar vocês. Obrigada.